When an initiator protein binds to the origin of replication, the strands are unwound by helicase, which breaks hydrogen bonds. Single-strand binding proteins keep the helix open and prevent it from forming secondary structures. A short string of RNA nucleotides called a primer, which is synthesized by the enzyme primase, provides a free 3' OH for DNA polymerase to add nucleotides. DNA polymerase III catalyzes phosphodiester bonds between the nucleotides. As the replication fork continues to move, the leading strand is continuously synthesized, while the lagging strand is discontinuous. Okazaki fragments are formed, and DNA polymerase I later replaces the RNA primer between the fragments and the lagging strand with complementary DNA. DNA ligase then joins the fragments. RNA polymerase catalyzes transcription after promoter sequences provide a signal to begin transcription. RNA polymerase adds nucleotides in a 5' to 3' direction. In the messenger RNA, uracil replaces thymine, and ribonucleotides are now used to form phosphodiester bonds. The 3 steps of transcription are initiation, elongation, and termination. In initiation, sigma factor binds to RNA polymerase to form a holoenzyme. The DNA is then unwound to form a transcription bubble. In elongation, sigma factor separates from RNA polymerase, which then moves in a 3' to 5' direction along the template strand, and adds nucleotides to the 3' end of the RNA. Termination occurs when RNA polymerase is released after reaching the termination region. After the primary transcript is formed, modifications are made. A capping enzyme adds a 5' methylated guanine cap that protects against degradation, flags for nuclear export, and can also act as a binding site for the ribosome. Poly-A polymerase cleaves the transcript at the poly-A signal at the end of the sequence and adds a poly-A tail. RNA splicing, which is catalyzed by a spliceosome, removes introns, which are sequences that are not expressed. In translation, the mRNA codons are translated to amino acids. Translation takes place on ribosomes that coordinate transfer RNAs, which each carry specific amino acids. Each tRNA has an anticodon that is complementary to an mRNA codon. A specific tRNA is covalently coupled to a specific amino acid, and base pairing with the charged tRNA directs the growth of the polypeptide chain. The start codon, which is AUG, initiates translation. The initiator tRNA carries methionine. The large subunit of the ribosome has an aminoacyl site, a peptidyl site, and an exit site. After the initiator tRNA binds, the next charged tRNA enters the A site. This tRNA has an anticodon that is complementary to the mRNA. The amino acid attached to the tRNA in the P site joins the amino acid in the A site, and a chain starts to form. As new tRNAs are added, the tRNA in the P site shifts to the exit site and is then released. Once the stop codon is reached, a release factor binds, and the ribosomal subunits, mRNA, and polypeptide chain are released.